Starting with the chapter 1, which is cell structure in syllabus 9700. This is the biology code for CIE exams. Now today we're going to handle the topic 1.2 of the chapter 1. The first topic is cell membrane. The cell membrane, we say, is partially permeable. Now why is it partially permeable is because it has these hydrophilic heads. Now the hydrophilic heads means that they are water loving, so they will allow water soluble molecules to pass. But the hydrophobic tails, this area does not allow any water soluble molecules or any ions or any polar molecules or any glucose. So this part, which is hydrophobic means water hater, phobia means fears. So this part will not allow any water soluble molecules to pass. So no ions can pass, no polar molecules, no glucose. Why? Because glucose is water soluble. So these will not be able to pass through the phospholipid bilayer. So that's why the other name for it is also the phospholipid bilayer, which of course you will study in chapter 4 again in great detail. So phospholipid bilayer. And why is it called a bilayer? Because you see it's got this part, and then there's this part, and then there is the middle part, which is the hydrophobic part, which does not allow any ions or polar molecules to pass through, but it will only allow small uncharged molecules to pass. Small uncharged, and that's very easy to remember because you can make a mnemonic of it, SUM. So small uncharged molecules can pass through it, and those small molecules are oxygen. Even water, though being a dipole, is a small molecule, so can pass through it. And the next heading is the nucleus, and it is a double phospholipid bilayer. So the nuclear envelope, the other name for the nuclear membrane is the nuclear envelope, as mentioned in your syllabus. So the nuclear envelope is a double phospholipid bilayer. So that means this is one phospholipid bilayer, and then there will be another one. So it's a double phospholipid bilayer which encloses the contents of the nucleus. And inside, of course, we have the chromosomes, which are found in pairs, which I'm showing you in different colors. So we have, in this cell, there would be 3 pairs of chromosomes. So 2n is equal to 6 and n is equal to 3. Another very important structure is the nucleolus. And here ribosomes are made. So this is the place where ribosomes synthesis takes place. Next is the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Now as long as you can figure out which one is which. Now the rough endoplasmic reticulum is the one which has got the ribosomes on it. So it's got these ribosomes which are sticking onto it, and the smooth endoplasmic reticulum, which is this part. Now this part does not have any ribosomes attached on it. Similarly here we've got this part of it. So all this is the part where you can see the smooth endoplasmic reticulum, which does not have any ribosomes, and the function of this is lipid synthesis. So this is very common found in the ovaries and the testes, where the hormones, the steroidal hormones, are made. The rough endoplasmic reticulum is the site on the ribosomes protein synthesis occurs. So all those cells where proteins are made, like cells in the pancreas where insulin is made, cells in the stomach wall where pepsin is made, cells in the salivary glands where amylase is made, they would have a larger component of the rough endoplasmic reticulum. So anything which makes proteins like lymphocytes would make antibodies. So these would be abundantly found in the lymphocyte. So rough endoplasmic reticulum and smooth endoplasmic reticulum. Next is the Golgi body. Now in plants, the Golgi body can also be called Golgi body, but it is sometimes also called the dictyosome. Now you must be able to pick it up. Now you see it's not an interconnected maze. It's like a series of flattened sacs. And we have the micrographs. This would be the micrograph which is showing it, and this is another micrograph which is showing the Golgi body has how it would appear. Now the functions of the Golgi body have to be very clear. It's basically modification and packaging. Modification and packaging. So it modifies the proteins. So wherever the proteins made, they were made on the ribosomes, and then they transported to the Golgi, and the Golgi modifies them, like for instance adding a glycogen part to it. So G for Golgi, G for adding a glycogen part to it. So we have some which are called glycoproteins. So the glycoproteins would have to go through the Golgi body to be able to be, so that the glycogen part can be added to it, and then sort of they are modified, and then they would be budding off, and they would be in the form of a secretory vesicle. They will be bud off like this one here, and then it would be of course by thrown out of the cell by exocytosis. So we go into more details as we go on to in the next few chapters. So Golgi body, modification, packaging, you must be able to pick it up how it looks on the micrograph, and in plants it has a different name, dictyosome. You must be able to identify the micrographs. Now as you can see, this one is the rough endoplasmic reticulum, which is also an abbreviation called RER, and this is the smooth endoplasmic reticulum, which is called the RER, and this is another one, SCR. So SCR and RER, these are the two which you have to identify in micrographs. So I mean, of course you can put it in the Google search and you can look for more diagrams, but these are few that I would like you to go through. So rough endoplasmic reticulum and the smooth endoplasmic reticulum. Smooth endoplasmic reticulum just looks like circles. Why? Because it is made of these interconnecting tubes, and when you cut these tubes, when you take a section of them, well, that's what you're going to see. You're going to just see little circles. And we have the mitochondria, and in the mitochondria what we have to understand is that the glucose, the food that you eat, like if you had bread or if you had rice or if you had potatoes, but all that is digested to glucose, but the body cells cannot use glucose. So the glucose is converted to the energy currency, which is ATP. ATP is adenosine triphosphate. So inside the mitochondria, ATP is made. Now we have to know the structure of the mitochondria. So this would be the outer membrane, which is a phospholipid bilayer. So outer membrane. Similarly, it has an inner membrane which is folded. Now it is folded because we need a whole lot of channel proteins which sort of fix here. And these channel proteins, now when you have a large surface area, so it's going to facilitate these channel proteins to be embedded. So outer membrane, inner membrane, and then the liquid part inside is called M for mitochondria, M for matrix, and then we have the 70S ribosomes. Now 70S ribosomes are the ones which are smaller than the normal ones which are found inside the cells. I mean, we'll be talking about it a little later. So mitochondria, outer membrane, inner membrane. The inner membrane is folded, It has proteins embedded in it. The liquid part inside it, or the gel part, is called the matrix. It also has its own loop of DNA. Now, if it has DNA and if it has ribosomes, that means it can make its own proteins. Next, we've got to understand chloroplasts, which are part of the plant cells, and they're present not in all plant cells, but in some plant cells which can photosynthesize. So chloroplasts can, of course, photosynthesize and make glucose by the combination of carbon dioxide and water, the results of formation of glucose molecule. But the important thing is that they can make ATP, and this process is called photophosphorylation. So the fact that the mitochondria can make ATP and the chloroplast can make ATP. Now the chloroplast structure has to be very clear. There is an outer membrane. So this is the outer membrane. Then we have an inner membrane, which is again a phospholipid bilayer. And then you have these structures in it which are called thylakoids. And this is one thylakoid. So there's the thylakoid membrane, and then there's the thylakoid space. And this is where photosynthesis takes place. Now one of these is called a thylakoid, but the stack of them is called a granum. Then it also has circular DNA, and it has the 70S ribosome, which is also found in mitochondria. So the mitochondria also is ATP is produced, and in the chloroplast also ATP is produced. Now you must be able to pick up its photomicrograph, which is something like this, and of course it has the starch granules in it as well, because when photosynthesis takes place, the glucose is immediately converted into starch and is stored. Now this is a very good comparison of mitochondrion versus chloroplast. ATP is made in both. The circular DNA is present in both. 70S ribosome are present in both, and they both have a double membrane. Of course, in the mitochondria, the outer membrane and the inner membrane is slightly different, is because the inner membrane is folded into these foldings are called cristae, and then in this, of course, the outer membrane and the inner membrane are not folded, but then you have the thylakoids in it, and then you have the granum, but both have a circular DNA and both have 70S ribosomes, and in both ATP is made. Next is lysosomes. Now lysosomes are actually a specialized Golgi vesicles, and they bud off from the Golgi apparatus, and sort of they'll pinch off from here, and this will result in a Golgi vesicle being formed. So Golgi vesicles will come off from here, and they will bud off, and if they contain hydrolytic enzymes, then they are given the name of lysosomes. Now basically they're present in cells to digest worn out organelles, like a mitochondria is now degraded or is not able to function properly, so that needs to be now digested. So digest, you can't sort of just throw them out, there's no dustbin in the cells, so they have to be digested, and then of course they're converted from the polymer to their monomers. Also lysosomes are used in phagocytosis, which you study in the last chapter, which is on immunity. Then we have to know the differences between cilia and microvilli. Now cilia and microvilli are two different structures. The cilia, you remember, are found inside the trachea. Now inside the trachea, the cilia actually move back and forth, and they move the mucus up towards the throat where it is swallowed. So the function of the cilia is to sort of sweep something, like sweep the mucus up to the throat. While the structure of the microvilli is just one cell, but its cell membrane, so it's just a normal cell. Now a normal cell would be like this, but no, this is slightly different. Now what has happened is that on one side it is thrown, the cell membrane is thrown into folds, and this of course increases the surface area, and you see a lot of these in the intestines. In the intestines we have microvilli which increase the surface area because the food that you ate, like for instance if you had roti, you had starch, Now the starch is digested to maltose, and then maltose to glucose. Now this glucose needs to enter this cell and then enter into the bloodstream. So this cell has to enter, and then it has to enter the cell. So on one side, to increase the surface area, you have these microvilli. So cilia is totally a different structure. It's like you say a hair-like. It's not actually hair, but it's a hair-like we say. And the microvilli is of course just the infoldings of the cell membrane. Next we have to study the different structures which are part of the plant cell. The first one, as we all remember from O levels, is the cell wall, which is made up of cellulose, and this is of course very thick. It's 500 times thicker than the cell membrane. So the cell wall is made up of cellulose, and inside this, with the blue color which I have used, is for the cell membrane. So the cell membrane is very, very thin, and it is very closely attached to the cell wall. Now in the cell wall, cellulose is of course the component which makes up the cell wall, and the cell membrane is made of the phospholipid bilayer. But we have these connections inside the one cell, and the adjacent cell has a connection, and this is called the plasmodesmata, this sort of cytoplasmic thread. Now we call this the cytoplasmic thread connecting to plant cells. We don't find this in animal cells, we only find it in plant cells. So these two cytoplasmic threads which are connecting are called plasmodesmata. Then we have the large central vacuole, and this is the large central vacuole which is present only in plant cells, and inside it is called the cell sap. So outside here we've got the cytoplasm. Inside here is the cytoplasm, and then the inside that is inside the vacuole is the cell sap. So the cell wall, which is made up of cellulose, the plasmodesmata, which are cytoplasmic threads. So if water moves through the plasmodesmata, is not osmosis, because it does not cross any partially permeable membrane. So if water moves through the plasmodesmata, it's not osmosis, because for the definition of osmosis, it has to pass through a partially permeable membrane. Now the last structure that we do in this topic 1.2 is the centrioles and the microtubules. The centrioles are only found in animal cells, and they are two rod-shaped structures which are at right angle to each other. So they're two rod-shaped structures which are found at a little, you can see them as right angles. You can see this diagram here which shows you the centrioles, plus this diagram here. Now these centrioles are made up of, we call it the 9 triplet structure. So they've got 9, these microtubules are arranged in threes, and this is called the 9 triplet structure. And each one of these is called a microtubule. So it's called a microtubule triplet. So the microtubules make the centriole, and these are important in spindle formation, which occurs in mitosis in animal cells. So plant cells do not have it, they have some other mechanisms which operates. So centrioles, two rod-shaped structures, and made up of microtubules. That will be the end of part 1 of topic heading 1.2. The next topic is the differences between plant and animal cells. Now, as we all know, there are some very, very clearly differences. Number one, the cell wall is present only in plant cells. Of course, cell membrane is present in both of them. So, cell membrane in both. Then cytoplasm in both. Nucleus is present in both. RER, SER, Golgi. The plant cell, of course, has a large central vacuole. Animal cells may or may not have, they have small temporary ones. Mitochondria is present in both, so you can see the mitochondria here and you can see the mitochondria here. And you can see the Golgi is present in both. And in some plant cells, of course, we will have the chloroplasts. Animal cell will have centrioles. Also, they will have the ribosomes. Now, the ribosomes which are present in the cytoplasm are the 80S ribosome. But the ones inside the mitochondria and the chloroplast has 70S. Similarly, the mitochondria also has 70S. We also should be able to identify and be able to label a photomicrograph. Now in this you can see some of it has been labeled, but the nucleus has not been labeled. So this area is the nucleus. And then you can see the chloroplast and you can see the cell wall, but you need to look at different diagrams to be really able to pick this up. And the vacuole membrane, of course, and the mitochondria. This is a micrograph of an animal cell, and because there won't be any, this will be the nucleus. And then of course we have the cell membrane, which has been labeled here as a plasma membrane, and would have the rough endoplasmic reticulum and mitochondria, lysosomes, Golgi apparatus, and this of course is the nucleus. And the next topic is prokaryotes. Prokaryotes are also unicellular. So prokaryotes are unicellular. So that means they will not have any tissues or any organs or systems. They're always just single-celled. And their main structure is that they have a cell membrane, which has also been called the plasma membrane or the cell membrane. It has cytoplasm. It has again 70S ribosomes. And it has a circular DNA which is in the form of a nucleoid, and this is the circular DNA which it has. Of course, it can be lying anywhere inside the cytoplasm. So the circular DNA and of course it also has the cytoplasm and the circular DNA, cytoplasm, ribosomes, cell membrane is the main thing which is present in every prokaryote. Of course, some prokaryotes would then have additional structures in it, which is could have some plasmids, could have a slime capsule outside it, and of course, yes, the important thing is that all bacteria have a cell wall, but this cell wall is made up of peptidoglycan. Peptide means a protein and glycan means a carbohydrate. So peptidoglycan. So a cell wall, cytoplasm, ribosomes. The cell wall has to have, of course, some bacteria would have a slime capsule outside the cell wall, and that's an additional feature. So all bacteria have a cell wall, cell membrane, cytoplasm, ribosomes, and the circular DNA. Prokaryotes also do not have any membrane-bound organelles. So no mitochondria, no chloroplast, no other membrane-bound organelles are present inside bacteria. The other most important feature which we have to talk about is the differences between, so all prokaryotes are bacteria. Now, animal and plant cells which have a proper nucleus are classified under a separate category, and they are called eukaryotes. So the differences between eukaryotes and prokaryotes have to be very clear, and you need to do a table and give me all the points which compare the prokaryote with the eukaryote. The last topic is that of a virus. Now, viruses have been classified as non-cellular. Why non-cellular? Because it's not even a cell. It does not have any properties in which it can be classified as a cell. The reason being that it has no functional, it does not function as a cell. So it only has a capsid. It only has a capsid which is made of proteins. So the outer structure which surrounds it is a capsid made of protein, and inside it there's nothing, there's no cytoplasm, and there can be a nucleic acid core which can either be a DNA or an RNA. That's it. So there's a DNA or an RNA strand, and there is a protein coat. In some viruses it has an outer phospholipid. So this is the outer phospholipid, and it has an outer phospholipid envelope. Now this outer phospholipid envelope is present only in some, it is only present in some viruses. So the outer phospholipid envelope present in some viruses, then a protein coat which is present in all viruses, and a DNA or an RNA strand, but no cytoplasm, no mitochondria, no chloroplast, no other organelles. And that is why it is also classified as, we wonder why we study it in biology, because it is not even living. But because it is able to reproduce inside another host cell, so we do study it. And of course it also causes a lot of diseases, and the one that we are suffering from now, the coronavirus, is a very good example of that.